Welcome to SOUMYA'S biology classes. I strongly believe that learning biology without understanding can be a torture, but with clear concepts it becomes fun and pleasure. In this lecture, I will be explaining the structure of a bacterial cell. Bacteria are included in the kingdom Monera of the chapter Biological Classification. Bacteria are single-celled or unicellular prokaryotic organisms. They are single-celled or unicellular prokaryotic organisms. Prokaryotic. Karyot means nucleus and pro means primitive. That means they have primitive nucleus. Primitive nucleus means that the genetic material is not bounded by the nuclear membrane, and membrane bound organelles such as endoplasmic reticulum, Golgi body, mitochondria, chloroplast, etc. they are absent. They are found in all types of habitats. They are found in all types of habitats such as bottom of oceans, below the icebergs, hot springs, inside or outside the body of other organisms, particulate matter suspended in air, etc. A bacterial cell is covered by a three-layered covering which is called the cell envelope. The outermost covering is called the glycocalyx. Inner to the glycocalyx, cell wall is present, and inner to the cell wall there is presence of cell membrane or the plasma membrane. Now let us see the structure of a bacterial cell. I am forming the outermost covering of the bacterial cell. This outermost covering of the bacterial cell is called the glycocalyx. It has a protective covering. Now this glycocalyx can be of two types. If it is in the form of loose sheath, thin, gelatinous, then it is called the slime layer. If it is hard, thick and rigid, then it is called the capsule. The slime layer is made up of polysaccharides, carbohydrates, and the type of polysaccharides that are associated with the slime layer, they are dextran, dextrin, and levain. The capsule is also made up of the same polysaccharides, but in addition to polysaccharides, amino acids are also present. And the type of amino acid which is present is D-glutamic acid. Where D represents dextrorotative basis of staining techniques, two types of bacteria are recognized: gram-positive bacteria and gram-negative bacteria. The bacteria that retains the gram stain, they are called gram-positive bacteria, and the bacteria that do not retain the gram stain, they are called gram-negative bacteria. Please keep this in mind that this capsulated condition is more common in gram-negative bacteria than in gram-positive bacteria. So this capsule is more common in gram-negative bacteria. Most of the pathogenic bacteria, they belong to the gram-negative group. This capsule protects the bacteria from the immune system of the host. Just beneath the glycocalyx, cell wall is present. Now I am forming the cell wall. It is present just beneath the glycocalyx. So this is the cell wall. The cell wall in bacteria is made up of peptidoglycan. It is also known as murin. Peptidoglycan is made up of modified sugars and amino acids. It is made up of repeating units of N-acetylglucosamine, NAG, NAG, and N-acetylmuronic acid, NAM, NAM. Just inner to the cell wall, cell membrane or plasma membrane is present. Now I am forming the cell membrane or the plasma membrane. It is present just beneath the cell wall. So this is the cell membrane or the plasma membrane. The cell membrane is made up of phospholipid bilayer plus proteins. It is made up of phospholipid bilayer plus proteins. The space between the cell wall and the cell membrane is called the periplasmic space. The space between the cell wall and the cell membrane is called the periplasmic space. This is the periplasmic space. The glycocalyx, cell wall and cell membrane together is known as the cell envelope. So the glycocalyx, cell wall and cell membrane together is known as the cell envelope. These are the ingrowths or invaginations or infoldings of the cell membrane, whereas these are the outgrowths or evaginations of the cell membrane. These ingrowths or invaginations or infoldings of the cell membrane, they are called mesosomes, they occur in the form of tubule, vesicle or lamellae. They increase the surface area and enzymatic content of the cell membrane. These mesosomes, they are of two types. The mesosome which is central in position is called the septal mesosome, and the mesosome which is lateral in position is called the lateral mesosome. This is the cytoplasm of the bacterial cell. In the cytoplasm, double-stranded circular DNA is present. In the cytoplasm, double-stranded circular DNA is present. And the region where this DNA is present is called the nucleoid. So this is the nucleoid. It contains DNA and associated non-histone proteins. So the nucleoid contains DNA plus associated proteins. Septal mesosome, they help in cell division. Whenever a cell divides, all the materials which are present inside the cell, they undergo duplication. So there will also be duplication of this DNA. And the duplication of this DNA is controlled by the septal mesosome. So the septal mesosome, they help in DNA replication and distribution of DNA to the daughter cells after cell division. When the cell is not dividing, this DNA is generally present in the center of the cell, but when it is about to divide, it gets attached to the mesosome for DNA replication. Septal mesosome is also involved in cell wall synthesis. It is involved in cell wall synthesis. Lateral mesosome contain respiratory enzymes. They are involved in respiration. They are involved in the production of ATP, just like mitochondria. Hence, the lateral mesosomes, they are also called chondroids. The lateral mesosomes, they are also called chondroids. In the 10th standard, you have studied that aerobic respiration involves 3 steps: glycolysis, Krebs cycle, and the electron transport system, or the ETS. Electron transport system, or the ETS, occurs in the lateral mesosomes. So the ETS occurs in the lateral mesosomes. Mesosomes, they are more prominent in gram-positive bacteria than in gram-negative bacteria. These are the outgrowths or evaginations of the cell membrane. So these are the outgrowths or evaginations of the cell membrane. Now these outgrowths, they are of 2 types. These smaller outgrowths, they are called fimbriae, and these longer outgrowths, they are called pili. Singular is pilus. Also called sex pilus or F pilus or conjugation pilus. Fimbriae, they are small, they are thin, and their number varies from 300 to 400 per cell. Their number varies from 300 to 400 per cell. Pili, they are longer, they are thick, and their number varies from 1 to 5 per cell. 1 to 5 per cell. Both pili and fimbriae, they help in attachment of the bacterium to the substratum, and both pili and fimbriae, they are made up of pilin protein. Now, this is the cytoplasm of the bacterial cell. In the cytoplasm, membrane-bound organelles like endoplasmic reticulum, Golgi body, mitochondria, chloroplast, etc., they are absent. But non-membranous organelles like ribosomes, they are present. This is the ribosome. And the type of ribosome which is present is 70S, where S represents sedimentation coefficient or Sedberg unit. Ribosomes, they are involved in protein synthesis. In eukaryotes, 80S type of ribosomes are present. In addition to the main DNA, small extra chromosomal double-stranded circular DNA may be present. In addition to the main DNA, small extra chromosomal double-stranded circular DNA may be present. This small extra chromosomal double-stranded circular DNA is called plasmid. So this is called plasmid. They are extra chromosomal DNA. They are extra chromosomal DNA because they are present in addition to the main DNA. And this plasmid, it is not dependent on the main DNA for its replication. These plasmid, they show autonomous replication. If plasmid is present in a bacterial cell, if plasmid is present in a bacterial cell, then the bacteria is said to be of positive strain, or F plus cell, or donor cell. If plasmid is absent in a bacterial cell, if plasmid is absent, then the bacteria is said to be of negative strain, or F minus cell, or recipient cell. Plasmid contains three important genes. Plasmid, they contain three important genes: F gene, R gene, and Col gene. F gene, also known as fertility gene, They are responsible for the formation of these sex pillars or conjugation pillars that facilitate the transfer of genetic material from the donor to the recipient bacterial cell by a process called conjugation. R gene, also called antibiotic resistance gene, they provide antibiotic resistance to the bacteria. For example, ampicillin resistance gene provides resistance against antibiotic ampicillin. Similarly, tetracycline resistance gene provides resistance against antibiotic tetracycline. Col gene provides resistance against colicin. They provide resistance against colicin. Colicin is an antimicrobial chemical produced by some strains of E. coli to control other strains of the same or related species. Now I am forming the locomotory organelle, that is the flagellum. একটা ব্যাকটেরিয়াতে ফ্ল্যাজেলাম থাকতেও পারে, নাও থাকতে পারে, বা অনেকগুলো থাকতে পারে। So this is the flagellum. The flagellum consists of three parts. This is the basal body, this is the hook, and this tubular part is called the filament. So this is the flagellum, and this flagellum, it is made up of flagellin protein. In case of eukaryotes, the flagella, they are made up of tubulin protein. In some photosynthetic bacteria, chromatophores are present. This is a chromatophore. These are pouch-like invaginations of the cell membrane. They contain photosynthetic pigments such as bacteriochlorophyll, carotenoids, etc., and are involved in photosynthesis. They are found in photosynthetic bacteria like cyanobacteria, that is blue-green algae, green sulfur bacteria, purple sulfur bacteria, etc. In some photosynthetic bacteria, membraneless vacuoles called gas vacuoles are present in the cytoplasm. They provide buoyancy to the bacteria. Storage bodies are present in the cytoplasm. They store reserve food material. So these are the storage bodies. Storage bodies are also known as cytoplasmic inclusions or inclusion bodies. So storage bodies, they are also known as cytoplasmic inclusions or inclusion bodies because they are included within the cytoplasm. Now these storage bodies can be of three types: PHB. PHB is polyhydroxybutyrate. They store reserve food in the form of fat. They store reserve food in the form of fat. Nowadays, PHB, they are utilized for the production of biodegradable plastics. Glycogen granules are present in the cytoplasm. Glycogen granules are present in the cytoplasm. They store reserve food in the form of carbohydrate. Volutin granules are present in the cytoplasm. They store phosphate reserves. Now, let us see some diagrams. This is a structure of a bacterial cell. This is the capsule. Innate to the capsule, cell wall is present. This is the cell wall. And innate to the cell wall, there is presence of plasma membrane or the cell membrane. This is the inclusion body. They store reserve food material. This is the pilus. These are the fimbriae. These are hair-like bristles that allow adhesion of the bacterium to the substratum. This is the cytoplasm of the bacterial cell. These are the ribosomes. They are involved in protein synthesis. This is the nucleoid containing DNA. And this is the extra chromosomal DNA, which is known as the plasmid. These are the locomotory organelles known as flagella. This is another structure of a bacterial cell. একটা bacteria section করে দেখানো হয়েছে. The outermost covering of the bacterial cell is called the glycocalyx. If it is in the form of loose sheath, thin, gelatinous, then it is called the slime layer. And if the glycocalyx is hard, thick, rigid, then it is called the capsule. Just innate to the capsule, there is presence of cell wall, and innate to the cell wall, there is presence of cell membrane. These are the ribosomes. These are the fimbriae. This is the main DNA or the chromosomal DNA. This longer outgrowth is the pilus, also called the sex pilus or the conjugation pilus. This is the inclusion body. And this is the locomotory organelle, the flagellum. And this is the cytoplasm of the bacterial cell, the cytoplasmic matrix. These are the fimbriae. This is the sex pilus that facilitate the transfer of genetic material from the donor to the recipient bacterial cell by a process called conjugation. These are the fimbriae and these are the flagella. This is a bacterial cell. This is the main DNA or the chromosomal DNA, and this is the extra chromosomal DNA known as plasmid. This is a diagram showing the transfer of genetic material from the donor to the recipient bacterial cell by a process called conjugation. This is the F plus donor cell containing the F plasmid, and this is a F minus recipient cell lacking the F plasmid. Pilus of the donor cell attaches to the recipient cell. Endonuclease enzyme cuts the plasmid DNA at a specific site called the origin of transfer, and one strand of the plasmid DNA is transferred from the donor cell to the recipient cell. The donor cell synthesizes the complementary strand to restore the plasmid, and the recipient cell synthesizes the complementary strand to become an F plus cell after conjugation. This is the donor cell or the F plus cell containing the F plasmid, and this is the recipient cell or the F minus cell lacking the F plasmid. Pilus of the donor cell attaches to specific receptors present on the cell wall of the recipient cell. This is the plasmid DNA. Endonuclease enzyme cuts the plasmid DNA at a specific site called the origin of transfer. This is the endonuclease enzyme, and this is the origin of transfer. The endonuclease enzyme cuts the plasmid DNA at a specific site called the origin of transfer, and one strand of the plasmid DNA is transferred from the donor cell to the recipient cell. The donor cell synthesizes the complementary strand to restore the plasmid, and the recipient cell synthesizes the complementary strand to become an F plus cell. Now the cell separates, and the bacteria, which was initially F minus, now become F plus after conjugation.